All right, team, welcome back. It is still Serene. We are today learning about bacteria. We're going to learn about bacterial cells, and then we're going to talk about an animal cell, but just your general eukaryotic cell and its components. So it's organelles. So today, cells and organelles, but we're going to start with bacteria. Bacteria. We know bacteria, right? Surely we've heard of bacteria. Have we heard of Mycobacterium tuberculosis? Tuberculosis, perhaps? Maybe? That's a bacterium. Bacillus. Okay. It's bacteria. We've heard of bacteria. They're like little guys. Like this general structure, maybe you've seen this, and it looks like, yep, it's sticking out. It's like, wow. But we'll get a better view of this in a second. Okay, so bacteria. What's going on? Bacteria, they can use chemical energy or they can use light energy. They have different nutritional modes. So we'll just say they utilize various nutritional modes because we eat food with chemical energy, and that gives us chemical energy. And then we use that to like go run around. So there's different ways to categorize different organisms. So we are heterotrophs. So we use food. So bacteria could be heterotrophic. So I'll just say hetero, or they could be autotrophic. So this defines our carbon source for metabolism. Defines for source of carbon for metabolism. So where are you getting your food? So heterotrophs, they use food, like, you know, how we eat like a sandwich. You know, you could have your bacteria colonizing your sandwich, your meat. Maybe that's why you don't leave your raw chicken out for 7 days. Generally don't recommend. But autotrophs use their atmosphere. So they'll use other stuff that's maybe not abundant in chemical energy. And so another category, so you have heterotrophs and autotrophs. We also have chemotrophs or phototrophs. And this is where we get our energy for metabolism. So we get our carbon from food for metabolism, but we also get our energy from our food. But some organisms aren't like that. So this is our source of energy. For metabolism. So that's the distinction between these two, auto versus chemo or photo. So carbon or energy. Okay, so what's the difference here? So for chemo, we're getting our energy from chemical energy. That's the same thing as using food. So we are chemoheterotrophs, and there are things like photoautotrophs. So you kind of compound, you bind it together. So we get our chemical energy from like an apple, and the apple also contains carbon, and that's where we get our carbon for metabolism. Phototrophs, photo means light. So it's getting its energy from light. So, for example, photosynthesis. It's synthesizing based off of light energy. That's pretty cool. So hetero is going to be using food. It's going to be some other source. Probably an autotroph it's going to fix carbon from the atmosphere, like carbon dioxide, right? And then a heterotroph could eat an autotroph, and then that's where it gets its carbon. With the chemotroph, it's getting its energy from chemical energy, something like stored in food. Or we could have a phototroph that's getting its energy from the sun, for example. Okay, so these are just our nutritional modes. Oh, I just said utilize various nutritional modes. Okay, so that's just a general insight to bacteria, how this is not just a bacterial thing, it's just kind of general, our categories of metabolism. So me, personally, as a human, I'm a chemoheterotroph. So photoautotrophs, an example of that is cyanobacteria. So that's like one of our first sources of oxygen, on Earth, is we had all this cyanobacteria in lakes doing photosynthesis, and that's how we got a bunch of oxygen in our atmosphere in the beginning. That's pretty cool. So bacteria can do all of these things. Me, personally, I could only be a chemoheterotroph. That's all I can do. But bacteria can do all of this. Bacteria can do it all. So they have a lot more habitats available to them as a result. More habitats unlocked. Because personally, if you put me in the mountains and I don't have any food to eat in the mountains, I'm gonna die. But maybe an autotroph can take the carbon from the atmosphere and then not die. So more habitats. Bacteria are pretty resilient. They're not as much of an extremophile as archaea, though, but we aren't really going to talk about archaea today, or ever. Someone will, not me. Maybe one day. Not today. Okay, so how does bacterial gene transfer work? Okay, so remember how last time we were talking about like binary fission? That's how we do some asexual reproduction. We're basically cloning ourselves. But there's other ways we can do gene transfer. Okay, so obviously evolution, it's good to have a variety of genes, because if you have a variety of genes in your population, some of those genes will be advantageous, and then some of your organisms with those good genes will survive and reproduce, and then we'll have organisms still. So genetic variation is really important for evolution. It's needed for natural selection to occur. So there's a couple ways we're going to get more genetic variation. And this can happen through vertical gene transfer. So this is not, we're not talking about binary fission yet. Right now we're just talking about ways to transfer genes from one organism to the next. So say you're a mommy bacterium, and then you have your DNA, and then you turn into two daughter bacteria. You're going to pass on your genes. That's like pretty simple. So vertical is like to your offspring. So your daughter cell, your daughter bacterium, it's going to have your DNA. That's almost like that's how that works. But this is like a major thing. This is one of the ways that we transfer genes. And as humans, we do that too. We don't do some of the other stuff. But this is like still really important. This is a thing that like how we have genes. And another way that we'll see vertical gene transfer is we can see, vertical gene transfer is like general how we know parent passes on their genes to offspring. But we also see vertical transfer of other things like mosquitoes. If they have dengue or Zika, they could, especially dengue, a mother mosquito that like contracts dengue, when it has its children, the children will also have dengue, which is kind of crazy. It won't be in their genome. It's not gene transfer, but it is a horizontal or a vertical transfer. Sorry, vertical. You can kind of see like this is vertical. Generationally, right? This is generation 1. And this is generation 2. So vertically across generations. That can even happen with mosquitoes. Like that's not gene transfer, but that's like another way that we have vertical transfer, and that's kind of crazy. Okay. So what about horizontal? We're talking about vertical gene transfer. You assume there's going to be a horizontal gene transfer. So, transfer. So your vertical gene transfer, that's going to be your offspring, right? It's happening from one generation to generation 1 to generation 2. And that's why it's vertical, You're kind of going in that straight line. But this one, instead of your offspring, it's going to be your friends. You're sharing your genes. And so you're, theoretically, obviously, things with shorter generation times, they're not going to be necessarily synced up with things that take longer to split and be in a new generation. But, we're all happening at the same time, right? Maybe your parent dies and then your daughter's cells are alive. But with horizontal gene transfer, you're here at the same time. So you'll have, for example, you'll have one guy and you'll have another guy. His genome looks like that. Their genome looks like that. Right? We got our circular DNA. And maybe you have a plasmid. You have these little balls of DNA. And you can trade. I can give you my plasmid, or I can take your plasmid. It doesn't have to be a trade. I can just give you mine, or I can just, take yours. But there's 3 ways we're going to do this. Okay, so your horizontal gene transfer is going to be between friends. So let's say I want to give you my little plasmid here. Bacteria have these things called pili, and they stick out, and you can reach over and be like, hey, I'm going to take your plasmid, so going to take the plasmid, or transmit them in some way. And so this physical transmission of DNA from one bacterium to another, that's conjugation. So sharing DNA through, and these are pili. I think they're called sex pili because it's DNA. That's kind of how that works. Through physical contact. We're reaching over to share some DNA. But maybe we want to share DNA not through physical contact, right? I assume that since I'm writing specifically through this way, there's another way, right? And that's going to be our transduction. So we can still share, we can get bacterial DNA from this cell to this cell without reaching over, and touching other people's DNA. Well, not people, bacterial DNA. And how this happens is you'll have bacteriophages. I'm going to go on to the next line. Bacteriophages, it's one word, bacteriophage, which is a virus for bacteria. And they'll take the DNA from one bacterium and put it in another. Transmit DNA between bacteria. And bacteriophages are pretty selfish, so usually what this does is it'll take DNA and then it'll implant it into a bacterium and then hijack it to make it produce proteins or more bacteriophages that is advantageous for the bacteriophage. But it can be advantageous for the bacteria because we're getting more genetic variation, which is good for natural selection. And then our third way, we have conjugation, transduction, now we have transformation. And what this is maybe I'm a bacterial cell and for some reason I eject a piece of DNA. I have a plasmid just sitting there. Transformation is when I just take that from my environment. I just, say a guy over here, I put this one out. I didn't want it anymore. So conjugation, we're sharing through physical contact. Transduction, we're still sharing, your bacteriophage is going to pick it up from someone and put it in someone else. And transformation, the bacterium will pick it up from its environment, take DNA from its environment. Pretty cool. So that's all of our lovely gene transfer among bacteria. Lots of different ways. So we have our vertical. This is pretty like self-explanatory. We're pretty familiar with the idea of vertical gene transfer. But horizontal transfer, or horizontal gene transfer, it's with your friends. You have conjugation, where it's physical contact. You have transduction, where it's bacteriophages. And you have transformation, where you take DNA from your environment. Pretty cool, if you ask me. And honestly, I feel like physical contact, bacteriophages, environment, those are like the big distinctions between these different sources, or these different terms: conjugation, transduction, and transformation. All right, so what is the thing that I was talking about before? I was talking about binary fission. What is that? That is not gene transfer. Okay, binary fission. So binary fission is just a bacterial cell reproducing. It's going to clone itself. It's not trying to get new DNA from anywhere else. It just wants to replicate who it is. So I'm just going to draw like a very basic diagram. So this is our circular DNA, right? We're going to start here. And we're going to start replicating our DNA. Because if we want two cells, we want to have two sets of DNA, right? So we're going to have our origin of replication, origin of where we're replicating our DNA. And I guess some textbooks might abbreviate this. It's ORIC. I don't know if you need to know that, but you can if you want. And so as you're replicating, you'll have your original set, and it'll start to like split out like this. Can you imagine like if I were pulling apart this bit, and like each set that I pulled was like a copy of another? That's kind of what's happening here. So this is kind of like the node at which it's connected. And so now our origin of replication, that would be like here, but it would also be like here because it's copied. And so the replication enzymes, as you're doing this, move out in both directions. So you have like your cell, and it comes to a point where it moves out all the way, where it's like two circles. And you're still like connected like right here maybe, like you haven't gotten all the way there. But now your origins of replication, they're like on opposite sides. And eventually, maybe this is better centered for you, eventually what you'll have is like two complete sets of circular DNA. And they should be exactly the same if the cell did it right, which lots of mutation occurs. So then we have this cell membrane and cell wall, right? I haven't drawn them distinctly, but we have, like, if this were this green with a cell wall, the membrane would be like right inside. So now we're going to have our two sets of DNA, and then we're going to have this area is like pinchen, because we want two cells, right? And this thing is going to form where it's like starting to create a wall and membrane here. So we're going to have a septum here. This is what a septum is. It's created by an enzyme called FTSZ enzyme. And so we're going to have like, and then our origins of replication are still here. And then our lovely chain going along eventually results in two bacterial cells. Yippee! Now we have two, which is excellent because this bacterium wants to overtake the universe, because that's how life works. So it wants to make as much of itself as possible, and so it's got to replicate. And now these guys can undergo binary fission. All right, so we have a general idea of how bacteria work. That's awesome. Now we can take a look into the cell. So we're going to look at a bacterial cell and a eukaryotic cell into the cell. And our eukaryotic cell is going to be an animal cell. but they're not that different from a plant cell. Or did I say? Yeah, an animal cell. Not that different from a plant cell. The differences that they do have that you need to know about, I will mention. Okay, so let's have a blob. A big old blob. Oh, maybe we should label this. This is bacterium. Bacterium is just one bacteria, you know? Okay, we have a big blob. So it's going to be kind of like, we're going to have like a thick layer of blob on this blob, okay? Okay, so what's going on? Bacteria have a thick, like, capsule that is sticky, and it adheres to surfaces and, like, your host, you know? Because if you're a parasitic bacterium, you want to stick to, like, I don't know, the throat of your host. So it's important to be nice and sticky. Sticky for adhering to surfaces and your host, very important. But if we were to take like a little, okay, hang on. I'm going to draw some spikes and it's all going to make sense one day. We'll get to it. But I just want to draw these spikes so that I can not draw lines through these spikes. Okay, so we have like layers going on here in this like thick membrane of this bacterial cell. It's not a membrane, it's like this, an outer layer, okay? So like on the outside, that outside layer, that's gonna be your capsule. And then we're actually gonna have lots of layers in a bacterium. You probably won't need to know all of them, but it is kind of good to know. So this layer here is going to be split up. We'll color this a different color. There we go. There's this thing here. Okay, so on the outside we have our capsule. It's sticky. So this capsule is like this whole layer here. It's sticky, it's kind of protecting your cell. And then here, this is your cell wall. And under your cell wall is your membrane, your phospholipid bilayer. So what does the cell wall do again? Remember how we had all that like extra support and structure with plants? Their cell walls keeps it from popping. So like mostly support. Does a little bit of protection some of the time. And then you have your membrane. We know what that does. Phospholipid bilayer. Bacterial cells are often characterized, all these words are in my way, with like a little tail. What is that? Okay, so this little guy is called a flagellum. And some cells, like animal cells, can have ways that we're doing locomotion. If I'm just like a little amoeba, I'm going to have, I think, cilia. And they're going to be like little bits that stick out, and they'll like be like little motors that'll help you go places. But flagellum, and you can even see flagella in animal cells, like a sperm cell. They have flagella. But bacteria, their flagella is for locomotion, similar to sperm cells. But they're very common in bacteria. Locomotion. You got to get around if you're a bacterium. You don't have like arms and legs to get you places, so you got to swish your little tail. Cool. Okay, so clearly there's probably going to be stuff in here. And you're going to have what I mentioned before, your circular DNA in this nucleoid region. Okay, so this is just a mess. Pretend it's connected. I don't think I remembered where I started that line, but theoretically the bacterial DNA is going to be one big circle, but maybe condensed and like scrunched together. Okay, this is the nucleoid. Remember that this is not bound by any sort of envelope, which we discussed in our last video about eukaryotic versus prokaryotic cells. And And as I talked about before, we're going to have little clumps of DNA just floating out and about, and those are plasmids. So this is like the majority of our DNA, like big DNA region. Plasmids are little pieces of DNA outside of chromosomes. Not outside of chromosomes, outside of your nucleoid. Which you can use for horizontal gene transfer. That's pretty cool. Not to be confused with plastids. Plastids are things like a chloroplast, and that is like a chloroplast or a leucoplast, a different type of like a little organelle in a cell. This is not an organelle. This is like a chunk of DNA. Plasmids with an M. Definitely got confused about that during when I first took bio. But little DNA chunks outside of the nucleoid. This is the nucleoid. These are our plasmids. A little different. Good for horizontal gene transfer. Okay, what else do we have going on here? We have these guys. Who are these? So these are fimbriae. Fimbriae. Awesome spelling. This is kind of similar to the capsule. It helps bacteria adhere to stuff. Stick to stuff. These look like pretty sticky guys. So, we don't have arms and legs. We don't have arms and hands to grip on. We gotta use our fimbriae. It's rough out there being a bacterium. I'm sure you know that I'm super experienced with the whole being a bacterium thing. You betcha. Okay, what else do we have? We have ribosomes, right? That's pretty cool. Actually, I'm going to use the darker green for this. I changed my mind. So ribosomes, that is where all of our proteins are made. Bacteria need proteins too. Just because they don't have a rough endoplasmic reticulum doesn't mean they don't get proteins. And you're like, I still don't know what a rough endoplasmic reticulum is. We are getting there. Okay, be patient. In the meantime, we're talking about ribosomes. We love ribosomes. This is where proteins are made. Proteins are made. Cool. That's your basic stuff about a bacterium. You probably won't even need to know all this stuff. Maybe you don't need to know about the capsule and all the layers. You definitely should know that they have a cell wall. What's it made out of? Do you remember? Peptidoglycan. That's what it's made out of in a bacterium. That's not very good. Look at that gap. Okay, let's fill that in. But yeah, just like general, what does a bacterial cell look like? Okay, let's move on to, instead of a bacterial cell, which is that prokaryotic or eukaryotic? It's prokaryotic. But this is just one kind of prokaryotic cell. Let's look at an animal cell, which is a eukaryotic cell. So animal cells, yep, type of eukaryotic cell. But this is going to be our basic blueprint of what we're going to be looking for in a cell. There's a lot of distinctions between an eukaryotic and prokaryotic cell. But one thing I want to draw that I didn't actually draw before is, so we have our fimbriae, right? Like these little pokey sticks. But sometimes we'll have like really long pokey sticks. And these will be those sex pili used for conjugation. So, pili, maybe it's pronounced pili, I don't know, used for conjugation. Let's share some DNA. That's another little guy that is cool. Okay, animal cell. Okay, so what's in our animal eukaryotic cell? What components do we have? Okay, I'm going to draw like a little, well, actually a pretty huge, like, circle kind of thing. It's not like a perfect circle because cells aren't like perfectly round. But this is like if we were to cut, you know? Like here we kind of cut open the cell. Here we cut it open like this. And we're going to have our nucleus. And nucleus is where all of our DNA is. So this is also going to get sliced. Let's say we sliced it like that. You know, like kind of like that. And it's like, oh, I'm cut open. Okay, so this is our nucleus. It has all of our DNA. Contains all the DNA. But it also has like this lump in the middle. This is the nucleolus. This is just kind of a region of the nucleus that contains all of the chromosomes with ribosome making information. So this is where ribosomes are assembled. Contains ribosome making DNA. I know lots of nuclear terms. We have nucleus, nucleolus, nucleoid. They are different. You, unfortunately, must remember this. Unless you mustn't. Maybe you're taking a gen bio class. Not gen bio, but like not a major bio class. Like you're just like a business major taking bio for funsies. Maybe you don't need to know what the nucleolus is. I don't know. I'm not a business major. Or maybe you're just watching this for fun, like on a Tuesday afternoon. Personally, that's pretty admirable. Also, we're going to have like a layer, an envelope, keeping it all like nice and together. And that's going to have holes in it. Or pores, if you will. So I don't know, we can like shade this in. Not very artistic representation here, but it's okay. Okay, so this will be our nuclear envelope, which I feel like is pretty self-explanatory. And this like hole here, that's a nuclear pore. And we can have stuff exiting from our nuclear pores. So if we're in our nucleus and we're making our DNA, we are also going to be making some RNA in the nucleus. And then that RNA is going to have to leave the nucleus and go to our rough endoplasmic reticulum. And the way it does that is through these nuclear pores. Okay, so I'm going to draw like it's kind of a weird looking thing. It looks kind of like this. It doesn't have to be precise. And it has lots of polka dots. And you're like, what are the polka dots? Well, we already drew the polka dots before. So this is our rough endoplasmic reticulum, to which I have mentioned very recently in the past. So this whole thing is our rough endoplasmic reticulum. And all those little circles are ribosomes. So what do you think we're going to be doing here in the rough endoplasmic reticulum? Well, here, ribosomes were used to make proteins. So I think this is where we make proteins. Makes proteins. So our rough endoplasmic reticulum contains a lot of ribosomes, and those ribosomes are going to be making proteins. And it uses that information from our nucleus, all that DNA, making RNA, and that RNA is traveling through the nuclear envelope and going to the ribosomes to make proteins. That's pretty cool. But connected to all of this, it's all connected. Not all of it, actually, but this is, I don't know if I would draw it like that. We have like, it kind of looks like sponges, almost like tubular sponges. Cause they're like, it's like, they have it's like a spout, you know, it's like they're open. Or at least that's how I've seen them drawn. It's like this like weird thing. And this is connected to the rough endoplasmic reticulum because they are both the endoplasmic reticulum. But this one is the smooth endoplasmic reticulum, because this one's rough. It has all these ribosomes, you know, sticking out, little bumps. This one's smooth. Endoplasmic. I don't know who came up with the name endoplasmic reticulum, but that's what we got. Okay, so we have the rough endoplasmic reticulum, we got the smooth endoplasmic reticulum, and the rough one has ribosomes, so it makes proteins. That's our rough endoplasmic reticulum. The smooth one, no ribosomes. That's why it's smooth. So what does that one do? I don't know, actually. I do. So that one, it's mostly producing lipids. And breaking down toxins. And also, well, I don't know why there's a comma there, I'll just write, and calcium storage. Storage of these calcium ions that the cell uses for whatever the heck the cell is using it for. Okay, so we have our nucleus, we have our rough endoplasmic reticulum, we have our smooth endoplasmic reticulum. Remember, our nucleus contains within it the nucleolus. It kind of looks like I'm saying the nucleus is just like this hole. The hole is the nuclear pore. The nucleus is this whole situation. That's the nucleus. The nuclear pore is this hole in the nuclear envelope. And the nucleolus is that area of the nucleus containing the ribosome making DNA. Pretty cool. And then DNA. Cool. That's what we got so far. What else do we have? Well, I'm sure you've heard of our cellular stars, the powerhouses of our cells. So the mitochondria are the powerhouses of our cells. When you learn in high school biology that the mitochondria is the powerhouse of the cell, that's wrong. One mitochondria is a mitochondrion, says the nerd, I fear. Okay, that's okay. Moving on. I'm a nerd. Mitochondria. So, mitochondria. So what does that mean? Powerhouse of the cell? Why? Because it makes ATP. We love ATP. That's the cellular currency, remember? It makes ATP. There's a whole song about it. I don't think it's about mitochondria, though. And it's also definitely not spelled that way. Okay, maybe there isn't a song about it. I'm sure there is a song about it. Okay, plays a big role in endosymbiotic theory because they have their own DNA. They have those, like, plasmids. Which is why we are like, hey, maybe this was its own little bacterial cell that we engulfed way back when. And they can reproduce on their own. Because they have their own DNA, they can reproduce on their own. Which is pretty cool. So what's going on here? Like, what am I looking at here? Because before it's like, oh, those circles, they are the ribosomes on the rough endoplasmic reticulum. So what is this? So basically, when you learn about cellular respiration, you'll understand you want the mitochondrial membrane to have as much surface area as possible. And basically, you have this membrane that's like folded over all this, a bunch to allow for like maximum surface area. And each fold, I'm just going to say like, each fold, like all these folds together are cristae. That's the fold. Those are the folds in the membrane of the mitochondria. Plural, mitochondria. And that just increases the surface area of your cell. That's pretty cool. Okay, what else can we talk about? There's so many things in an animal cell. So this is one of those things that's kind of like specific to animal cells. You might not be asked about this, but they kind of look like pasta, like they're kind of like round and then they're like kind of like, I don't know. But you'll see them when you get to mitosis. These are centrioles. They're really important for mitosis. They like separate your chromosomes and stuff, and you'll learn about that later. But they're also they play a big role in determining where the stuff in your cell is. So they determine where things are. Okay, so those are centrioles. That's kind of all I have to say about that. And then we have a couple like circles of the cell, a couple spheres going on. So I'll put one here. They're just kind of all over. They'll go through like your, oh, we need to talk about her. Oh, where am I going to put her? Okay, well, they'll go through your Golgi apparatus, which I don't know where I'm going to fit, but it'll be fine. But like these little orbs. We'll cut one open. This one's going to be open. These are vesicles. It's just like a capsule that's moving stuff around the cell. So it's just a capsule transporting stuff. Cellular materials that need to go from one place to another. It'll get packaged up from the Golgi apparatus and need to be sent somewhere else. So what the heck is a Golgi apparatus? We're going to put her here, which is going to be super easy. Okay, so she looks like kind of like a pancake stack. Like folded over. Like if I were to like have like a bunch of like pancakes kind of together. Kind of. What's going to happen is you'll have stuff maybe go from the smooth endoplasmic reticulum and it'll go through the Golgi apparatus and it'll get like packaged up in a certain way and then it'll go out. So like this way maybe. It'll just be like stuff and it'll be sent in and then like sent out like as a vesicle. Maybe I should have drawn a vesicle there. Maybe I should have drawn the arrow pointing to the vesicle we already had. But now there's a vesicle there. So this is our Golgi apparatus. We're going to draw, well, maybe we should write it first. So Golgi apparatus. So it's packaging stuff, right? It's apparatus. It's getting stuff prepped to go somewhere else and be in the right form. So we call this the UPS of the cell. It's our UPS. Because it's packaging stuff up and it's getting it sent places. It sorts and sends off vesicles. So the end where it's receiving something from, for example, the smooth endoplasmic reticulum, this is going to be the cis face. Cis face. It's going to be receiving. And then trans means to go across, right? Trans, that root word means across. So as it goes across, it'll be going through the trans face. There's not much space here. But that's what that is. So this is the cis face. That's the trans face of the Golgi apparatus. But the main thing you want to know is it's the UPS. It sorts and sends off vesicles like the UPS. That's what they do, if you didn't know. Okay, so one type of, I wouldn't say it's like a type of vesicle, but it basically is. It's like another sphere full of stuff in your cells. You'll have this thing called a lysosome. Remember a while ago when I was talking about water in cells and how if you have too much water in your animal cell, you can have lysing, where it pops. Well, the lysosome makes that happen, and you'd be like, why would you want to pop? Okay, so what a lysosome is it contains digestive enzymes. I'm going to put it over here. No, we can fit them. Digestive. You should really have your lysosome, all those words, like full inch over, but I didn't do that. And that way, if you want, you can initiate apoptosis, which is programmed cell death. So they call that the cell suicide, because what happens, what happens is maybe you're an awesome cell and everything is going great, and then one day you mutate and you are crazy, and the other cells are like, guys, get this guy out of here. So they'll send a message to the cell and they're like, okay, you have to die. And what will happen is these lysosomes full of digestive enzymes, which are really important for, like, say you make something in your cell and then you use it and now you need to break it down. You need all those, like, important building blocks for whatever it is you created that you don't need anymore. Maybe you're, like, eating something and it needs to break down the thing that you're eating. Those digestive enzymes are important. But another thing the lysosome does is, if it's told to perform apoptosis, important term, or programmed cell death, then all of those digestive enzymes will be released and just released into the cell, and the cell is going to die. So that's important. Lysosome. And we also have our centriole, very important in mitosis, but also determining where things are. Digestive enzymes. Mitochondria makes ATP, and they have their own DNA. That's why they can reproduce on their own. Big part of endosymbiotic theory. Also, you can have peroxisomes. Notice how, this is my lysosome. They're all kind of just like circles. They're spheres containing stuff. So they don't look that different. So, like, you probably won't be asked to, like, look at a slide and, like, is this a lysosome or peroxisome? Because you probably won't know. But we should know, like, what is a peroxisome? What is a lysosome? What do they do? So peroxisomes, hydrogen peroxide, it has hydrogen peroxide, H2O2, baby. And they are really important for, in plant cells. So, implied by the name, hydrogen peroxide, peroxisomes, got that hydrogen peroxide, H2O2. And in plant cells, these are really useful. They're in animal cells as well, but they're very useful in plant cells because plant cells will do a thing called photorespiration. You'll learn about that later, but it's bad. You don't want to do that. And basically, it uses up a bunch of good stuff and makes bad stuff, and peroxisomes kind of like salvage what you did there to kind of recover it. So that's important in plant cells. In animal cells, H2O2, you're going to be undergoing, like, oxidative reactions because you have your oxygen. So it's just important for, like, oxidative reactions. Because you got that hydrogen peroxide. Cool. That's basically your tour of the cell. Lots of stuff. Very exciting. Okay, your peroxisome. You got your H2O2, oxidative reactions. That's cool. You have your lysosome. So they're both the zomes, They're both like sacs of stuff. This sac has like hydrogen peroxide and stuff in it. Lysosomes have digestive enzymes. They can initiate lysis. Lysis, because it's a lysosome. That's your apoptosis. The apoptosis is like programmed lysis. Lysis is when the cell, like, explodes or just, like, dies. So your lysosome can make that happen if your cell is going crazy. And you'll learn a little bit more about the cell cycle and why you would want a cell to die if it's going crazy. What does that mean? You have your nucleus, all your DNA, your nucleolus center with all the ribosome making stuff. Endoplasmic reticulum, all that information goes out through the nuclear pores of the nuclear envelope, through your endoplasmic reticula, not too sure. The rough stuff makes the proteins. The smooth stuff makes the lipids, stores some calcium, breaks down some toxins, but most importantly, we produce those lipids. Our mitochondria make ATP. Basic overview of the cell. Golgi apparatus has the cis face here, and then once it does all of its stuff, it sends out vesicles through the trans face. And what is the difference between an animal cell and what you might need to know from a plant cell? The only main difference is centrioles are only in animal cells, and plant cells have a cell wall. So this is not pictured here. The cell wall, remember, is really important for having that hypotonic environment, turgor pressure, pushing back on the cell, keeping it from popping. And also, plant cells will have chloroplasts. You'll learn more about chloroplasts when we get to photosynthesis, but that's what they enable. They have chlorophyll to do photosynthesis, turn light energy into chemical energy. And just a review of our bacterial cell, we got our nucleoid with all of our DNA, big circular DNA, and then we'll also have little tiny pieces of plasmid DNA, which can be traded through horizontal gene transfer. If you use your sex pylide, then it'll be conjugation, but if you just pick it up off the floor, then it'll be transformation. And if a bacteriophage injects it into you, then that's transduction. Little review there. Flagellum for locomotion. We have cell walls for support. Remember, that's why we don't have animal cells with cell walls, because they're chilling. They like an isotonic environment. So if you have like a bowl of water and you want to put an animal cell in it, you have to like put in the exact right amount of salt that should match your animal cell solute content. Kind of interesting. So yeah, that's our little review. Bacterial cells. What does that look like? How does that compare with an animal cell? All these organelles are very, very important. You should definitely know just about all of these. You might be able to give or take a peroxisome, but everything else on here, I don't know, you might be able to give or take some of the nuclear elements, depending on the class you're taking. But if you're taking Gen Bio, you gotta know basically all this. So get studying, get memorizing. I hope you drew this. Diagrams. Best part of biology, guys. Unless you disagree, in which case you'd be wrong.